Hi everyone. Today I'm going to walk you through one of the projects we have going on in my group at Wayne State University regarding the microfluidic measurements of protein biomarkers from exhaled breath. Now, I think everybody at this conference really understands how powerful exhaled breath is as a clinical sampling medium. And whereas most people are looking for volatile organic compounds as biomarkers from disease, my group is developing complementary tools to look for protein and nucleic biomarkers as well. So specifically, what we're doing is developing assays using beads. So figure 1 shows you one example of a magnetic bead. So the idea is we take these beads and we functionalize them with some sort of biorecognition element. So like a capture antibody if we're targeting proteins, or a DNA probe if we're looking at nucleic acids. Now, what we can do now is take some tens of thousands or hundreds of thousands of these beads and incubate them with less than 100 microliters of some sort of biological sample like exhaled breath condensate. So what will happen now is you have such a high surface area of capture elements that you can really efficiently extract out target analytes from the sample. So once you have all of these beads that have captured sample or not, you can then load them into an array of microwells, where each microwell is designed to hold only a single bead. You can then subject this to some sort of amplification, either PCR or ELISA. And the idea is that if an individual bead in an individual well has captured a target analyte molecule, it will exhibit fluorescence after amplification. So the local dye signal inside that well is actually really high. So now rather than trying to measure really dim, low fluorescence analog signal, instead we're simply going to count the number of wells that are glowing after amplification, because that percentage of active glowing wells is directly proportional to the analyte concentration in our original sample. So you can see in figure 3 what one of our microfluidic devices look like. The SEM inset just shows beads loaded into wells, and you can see most wells contain a bead, but not all, and that's okay. Because before PCR, what we do is take an image where in figure 4, all those little white dots are just beads. So you can see a little bit of bead autofluorescence before PCR. But now after PCR, you can very clearly see which wells lit up, meaning which ones contained a copy of the target analyte. So we have developed this technology for both DNA and proteins. Today, I'm just going to show you some data from the protein assays, just because I think it's a little bit more interesting. So we chose 3 model cytokines as target proteins. And if you look at the calibration curve of active wells as a function of concentration, you can see how low we can really detect. The limit of detection for our digital protein assay is, 4 femtomolar for GM-CSF. It's 30 femtomolar for interleukin-6. So these are quite low compared to any sort of conventional method that you would use to analyze these proteins. And the limit of detection is just largely based on how good your capture antibody is. But analyzing proteins individually is not interesting. The real power of diagnostic medicine really comes in multiplexing. So trying to measure multiple analytes at the same time from the same sample. So what we do to achieve this is by encoding different beads with different amounts of an encoding dye. So we designed 3 different bead populations that do not overlap in fluorescence, so that when you take beads that are encoded with each of the 3 dyes, that are targeting 3 different proteins, you can mix them together and put them into the same device for analysis. So that brings me to the exhaled breath condensate analysis. So we collected EBC from healthy volunteers, and one of the very first things we did was just to screen for saliva contamination, because it's important to us that the biomarkers that we're looking for originate from the lungs and not from the saliva. We just quickly showed that a saliva sample falls right in the middle of our calibration range, but in an inhaled breath condensate sample, we did not detect any amylase, which suggests that there is no saliva. So all of our sample really started from the lungs, and that's where our biomarkers originate from. So from there, we took the beads that we had developed, so 3 different bead populations, 3 different levels of encoding dye with 3 different capture antibodies. All right. So each of those 3 bead populations was mixed together and put into one EBC sample. So you can see in the decoding wavelength which beads were for the high intensity, medium intensity, and low intensity populations. After that, we ran the PCR assay to amplify captured target proteins. So by then taking an image at the assay wavelength, you can see which wells exhibited signal after amplification. So then overlaying the decoding image on top of the assay image, we essentially treat this multiplexed assay as 3 individual assays. So all of the bright beads were quantified for their assay signal. All of the medium beads were quantified for their assay signal. Same with the low beads. So with this strategy, we were able to precisely quantify 3 different cytokines from the exhaled breath condensate sample. And they range from between 5 femtomolar to 400 femtomolar. Now note that this could not be achieved using conventional ELISA, whose limit of detection is 500 femtomolars. So with this innovative microwell array approach, we were able to analyze 3 different cytokines from an exhaled breath sample that could never have been done using conventional technology.
The presence of volatile organic compounds in human breath and the ability of exhaled air analysis to identify diseases has been studied with both targeted and untargeted approaches. While targeted approach is hypothesis-driven and relies on the quantification of predetermined volatile organic compounds, untargeted approach is based on VOC profiling and can provide information for hundreds of different substances. In this context, the objective of the study was to compare the efficiency of target and non-target breath VOC analysis in the discrimination of lung cancer patients from patients with other respiratory diseases and healthy controls. The population sample consisted in 3 groups. We approached patients with pathological findings on chest computed tomography who underwent bronchoscopy, biopsy, and cytological or histological analysis. Thus, 2 groups were formed. Group 1, which were patients diagnosed with lung cancer, and Group 2, not diagnosed with lung cancer, while a 3rd group was formed, consisting of healthy controls. Sampling was conducted with the use of Tedlar bags. Pre-concentration and extraction of the analytes was achieved with solid-phase microextraction, and instrumental analysis was performed with gas chromatography mass spectrometry. In this figure, the median concentrations of the selected organic compounds in all participants are presented. Acetone and isoprene were the most abundant compounds in human breath. 2 propanol and 1 propanol were also detected in high concentrations. Regarding aromatic compounds, the highest concentrations were observed for toluene, but significantly lower levels were measured for styrene, ethylbenzene, and benzene. Aldehydes were also present in the vast majority of the samples analyzed. This table presents a comparative analysis of VOC concentrations between population subgroups. 5 compounds were found to differ significantly between patients and controls. None of the investigated volatile organic compounds differed significantly between lung cancer patients and patients not diagnosed with lung cancer. As shown in this diagram, the median concentration of monoaromatic compounds were higher in patients before and after subtracting ambient air concentrations. Observed variations in the concentrations of monoaromatic compounds may reflect a general impairment of pulmonary function, and these biomarkers are obviously not specific to lung cancer. 1 and 2 propanol concentrations also differed in subgroups, and for these substances, breath substracts were negative, indicating higher levels of ambient air than in breath. 1 and 2 propanols in human breath are endogenous or exogenous origin and are rapidly absorbed and metabolized to the corresponding aldehydes, acetone and propanol. Thus, negative corrected concentrations are observed. The reaction is catalyzed by alcohol dehydrogenase. Untargeted analysis was performed with the online bioinformatic platform XNS Online. After peak detection, alignment, and retention time correction, online statistical analysis was performed to identify informative peaks.
Hello, my name is Alp Kilic. I'm from Turkey. I'm a PhD candidate at Gebze Technical University Department of Physics and working on research and development of gamma-resistive gas sensors with sensor group. I would like to present one of our study about the breath biopsy research. Our study is related with fabrication of semiconductor metal oxide gas sensors for breath biopsy applications. Breath biopsy has attracted much attention for disease monitoring due to its non-invasive and fast diagnostics ability. Breath biopsy has been realizing by the evaluation of collected breath samples from human exhaled breath. Several substances in human exhaled breath are considered as biomarkers, which are correlated to varying diseases. For example, acetone concentration in the human exhaled breath is related with diabetes. Also, hydrogen cyanide and ammonia are related with cystic fibrosis and kidney diseases, respectively. To achieve accurate diagnosis, minimum detection limit for a sensor should be in the range of sub-ppm or ppb level for breath biopsy applications. Detection of these low concentrations is a major challenge in the development of a breath analyzer system. The emphasis is placed on nanostructural semiconductor metal oxide-based gamma-resistive gas sensors to meet these requirements due to their adequate reactions with biomarker molecules. Also, nanostructural morphology provides high surface to volume ratio and high sensitivity. The basic principle of gas sensing mechanism of a metal oxide material is based on chemical reactions between gas molecules and surface of the material. These chemical reactions change the conductivity of metal oxide, and this change is named as sensor response. You can see the typical conductivity change and time graph of metal oxide material in this figure. From this graph, we can observe many parameters about metal oxide gas sensors, such as sensor response, response time, recovery time, sensitivity, selectivity, or reproducibility. In this study, we fabricated titanium dioxide, copper oxide, and both non-oxide nanostructures on aluminum substrate with a very simple and environmentally clean hydrothermal route. We coated gold interdigital electrodes on the fabricated samples to transforming of sensor device. Then tested against acetone, hydrogen cyanide, ammonia, and varying interference gases such as nitric oxide and toluene. In this figure, you can see our sensor test system that is including electrometers, temperature controller, gas chamber, and a gas generator. After the sensor tests, we used scanning electron microscopy technique for investigation of surface morphology. Titanium dioxide and copper oxide were grown as nanorods, and the wolfram oxide was grown as nanoflakes as seen as EM images. Each of the non-structural metal oxide was homogeneously covered on the substrate. While titanium dioxide nanorods are average 1 micrometer in length, copper oxide nanorods are average 250 nanometers. After the fabrication and characterization, we performed biomarker sensor tests according to our sensor test procedure. At the first step, we tested all sensors against 1 ppm hydrogen cyanide, ammonia, and acetone at the temperature range between 50 Celsius and 300 Celsius for investigation of optimal operating temperature. Optimal operating temperature was identified as 200 Celsius for wolfram oxide and 250 Celsius for titanium dioxide and copper oxide, as seen in this graph. Then, as second step of our sensor test procedure, we scanned varying concentration of each cases from high level to low level. After this step, we observed that minimum experimental detection limit of sensors as shown in sensor response peak graphs. As seen in these graphs, each of the sensors showed a clear response peak under the biomarker ambience. Experimental detection limits for this study are 125 ppb hydrogen cyanide for copper oxide, 450 ppb acetone for titanium dioxide, and 1 ppm ammonia for wolfram oxide. And all these concentrations are disease level of cystic fibrosis, diabetes, and kidney diseases respectively. Also, you can see in this graph sensor response of each sensor against varying interference gases at the optimal operating temperature. In here, especially wolfram oxide showed a dramatically ammonia sensing performance with excellent sensitivity and selectivity. And finally, as a conclusion or a general overview, we fabricated 3 nanostructural metal oxide-based gas sensors with a very simple hydrothermal technique, then performed morphological characterization and investigated their biomarker sensing ability. Each of the sensors are good candidates for further level sensor array applications due to their low cost, easy fabrication, and their highly sensitive and selective sensing performance against biomarkers for early diagnosis of kidney diseases, cystic fibrosis, and diabetes. And thank you very much for your attention. If would you like to ask any question or detailed information about this research, please don't hesitate and contact me. Thank you.
Hello everyone, my name is Mohammad. I'm going to give an overview of a recent finding of breath biomarkers of insulin resistance from a cohort of pre-diabetic Hispanic adolescents in San Antonio, Texas. In background, an estimated 1.26 billion adults have insulin resistance. Insulin resistance is a metabolic condition associated with an imbalance between insulin and glucose metabolism. This is also strongly associated with the development of cardiometabolic disease. According to a meta-analysis based on 18 population-based studies from 13 countries, on average 25% of children among 6 to 9 years of age have insulin resistance. There are multiple blood-based surrogate tests exist to measure insulin resistance, but most of these require fasting for 8 hours, multiple sampling, and most importantly, only performed after the onset of diabetics. Our previous studies in a cohort of non-human primates showed that animals develop insulin resistance by 3.5 years of age and cardiac defect by 5 years of age on a nutrient-restricted diet. Axial breath VOC signature discriminated between these with cardiometabolic dysfunction compared with control. Based on this info, we hypothesized that the axial breath VOC will correlate and predict cardiometabolic status in humans, and our goal was to identify a small set of VOC biomarkers in a pre-diabetic population that are important for early detection of cardiometabolic disease. In method, the participants of this study were recruited through the Health and Weight Management Clinic at Children's Hospital of San Antonio. A total 28 Hispanic adolescents were recruited in the study using an inclusion criteria of age 13 to 17 years, BMI percentile more than 95, and from Hispanic ethnicity. The collected blood samples were analyzed for total sugar, lipid, and liver profile. The insulin resistance were calculated based on HOMA-IR. Breath sample were collected using Reciba device, and the medical grade air was supplied to remove any background contamination. Total 4 replicates were collected from breath sample from each participant. Comprehensive two-dimensional gas chromatography hyphenated with time-of-flight mass spec was used for breath analysis using TD100 thermal desorption unit. Data cleaning and processing was performed using Chromatoph software. Additional data analysis pipeline was developed and performed in R statistical program. In result, data processing pipeline removes unwanted variation of the breath dataset. In figure 2, we observed no absolute clustering within breath sample in green and medical air sample in red. This is a proof of data normalization and comparability. In figure 3, we have used random forest regression machine learning method for feature selection. 2/3 of samples were used for develop a training model, and remaining 1/3 were used for validation. The top 10 features based on the importance measure were selected and used for the prediction of HOMA-IR. As shown in figure 4a, we observed a strong correlation with breath model predicted IR and experimental HOMA-IR. We also observed a strong correlation in the breath-based IR with the fasting blood glucose and insulin, although this data is not shown here. This correlation in 4a is a combination of 10 metabolites, but in figure 4b, we identified the metabolite limonene by itself clustered between normal insulin resistance and borderline insulin resistance. This pilot study shows feasibility of cardiometabolic breath signature in pre-diabetic children. We identified a signature of 10 breath metabolites that highly correlated with IR. IR is also known to strongly associated with non-alcoholic liver cirrhosis. One of our identified metabolite, limonene, has been reported by others in literature as important for liver cirrhosis. This is also confirmed by our clinical measurement that the children in this study already have early sign of liver damage. Our breath signature can be developed further to monitor and track the progression of cardiometabolic disease. Thank you for listening, and please let us know if you have any comment or suggestion.
Hi there, thanks for calling in on this poster. I just wanted to make 2 main points so that you can take them away. I think the first is that by using stable isotopes, you can achieve really high specificity for a limited number of reactions. And secondly is that even in the case of really kinds of hairy pathogens, so this is a select agent and all work is done at BSL-3, there are usually ways of getting gas samples out. In this case, we had to absorb it onto Sodasorb so we could autoclave the sample out. Okay, I hope you enjoy the rest of your meeting. Bye.
Hello, this is Danling Wang, an assistant professor from the Department of Electrical and Computer Engineering. I'm from the North Dakota State University. In this proposal, I'm going to present a novel breath acetone sensor based on a newly synthesized nanocomposite for the purpose of the diabetes prevention and monitoring. The major motivations of this device are based on the two factors. The first factor is about the current U.S. diabetes situation. Each year, there's more than 80,000 patients died due to this disease, and the number of diagnoses and undiagnosed diabetes has increased a lot, in particular including some young children. The cost for the diabetes diagnosis and the monitoring is also very expensive. The second reason is the current technique for the diabetes diagnosis and monitoring mainly based on the blood test. So this type of test is very invasive and inconvenience and high risk of the infection. And some testing is very expensive because you need to rely on the expertise to operate the machine and do the data analysis. The fundamental of this device is based on these two working principles. The first working principle is breath acetone was one of the breath volatile organic compound is directly linked to the metabolic activity in human body and in diabetics, and so it can be a biomarkers for the diabetes diagnosis and monitoring. The second principle is the research indicate about a close relationship between the breath acetone and blood glucose. So in particular for the type 2 diabetes, they have almost positively linked together. So you can use the measurement about breath acetone to indicate about the blood glucose level. The device we developed with features of the low power, low cost, non-invasive, non-toxic, and accurate, and also will be based on the Internet of Things, make the device can do the in-time communication, real-time monitoring, and remote sensing. This device is has been made based on two new sensing materials. One is a one-dimensional nanowires we call KW. This is a material with unique ferroelectric property to interact with acetone. Another is two-dimensional nanosheet we call the titanium carbide maximum. This material has very large surface and interface area and a tunable surface functional group. It can make the overall sensing material composite can selectively and sensitively to detect acetone. Our results indicate a certain ratio about KW and titanium carbide can indicate the highest response to the acetone, even when the acetone level is 2.86 parts per million, and much less interference from the humidity, and can give a much stable performance with variable humidity condition. The sensor device is based on a chemi-resistant response. It is like this picture shows when the acetone attached on the sensor film surface, they will cause a resistivity change. We can measure the resistivity change can indicate about concentration of the breath acetone and then blood glucose level. In the future, the device will be enabled about wireless transfer and deep learning to make a smart decision and real-time communication. So the device will be low cost, high accuracy, low power, and IoT enabled device. This research has been supported by the NASA and NDFSPR and the local hospital, Sanford, and NDSU. Thank you.
My name is Daria Slefarska. I am PhD student at Jan Kochanowski University in Kielce, Poland, and I would like to introduce you to the following topic: the volatilomic signature of human gastric cancer cell lines HGV-27 and CLS-145. Analysis of volatile organic compounds released by human body provides an emerging approach for cancer screening. Human volatiles can have a systemic origin or stem from exogenous sources such as environmental exposure, diet, or microbiota activity. These compounds form specific biochemical signatures which can be altered by abnormal processes occurring in the organism, including cancer. These alterations can stem from changes in enzyme activity, modification of proteins, or activation of genes. The detection of these changes via analysis of volatile organic compounds emitted by breath, skin, and other bodily fluids provides a unique opportunity to screen or monitor various diseases, including cancer. This approach suffers, however, from a number of limitations. The main unresolved issue here is the often poor biochemical understanding of the origin, behavior, and metabolic fate of volatile biomarkers in the human organism. In vitro studies involving these cell cultures can partially address this issue. The main goal of this study was to determine the volatile metabolomic signatures of two selected gastric cancer cell lines, HGV-27 and CLS-145, and one normal line, HSEC, and detect possible differences between their fingerprints. More specifically, this involves determination of volatiles that are produced and used through metabolic processes of the cells, and identifying changes in the volatile organic compounds production that are caused by the cancer. Within this study, two cancer cell lines were investigated. The CLS-145 cell lines was derived from fragments of a gastric papillary adenocarcinoma of pars cardiac resected from a female therapy-naive patient. The HGV-27 cell lines was established from a gastric cancer patient with a histologically diagnosed carcinoma. Non-carcinomia human stomach epithelial cells, isolated from normal human stomach tissue, were used as a control line. For this study, headspace needle trap extraction and as the pre-concentration method and gas chromatography with mass spectrometric detection have been applied to respectively capture and analyze the headspace above the cultivating medium and cells in the cultivating medium. Altogether, 12 sets of cultures containing each 3 cell cultures and 1 medium without cells were prepared. Amongst the volatiles detected, 27 showed differences in their headspace concentration compared to those above the cultivation medium only. 12 species were found to be consumed and 10 were produced by all lines under study. Of consumed volatiles, there were 8 aldehydes, 3 heterocyclic compounds, and 1 sulfur-containing compound. Whereas the production of volatile organic compounds embraced 7 methyl ketones, 3 esters, 3 alcohols, toluene, and 2-methyl 3-methylthiofuran. HGV-27 cancer cell lines were found to have significantly altered metabolism in comparison to normal gastric cells and CLS-145 cell line. This was manifested by the increased production of methyl ketones containing an odd number of carbons. There were also 3 ketones produced exclusively by the line, namely 2-undecanone, 2-tridecanone, and 2-heptadecanone. HGV-27 produced also toluene. Another interesting feature of the HGV-27 volatile footprint is the lower production level of alcohols and esters. The CLS-145 cell exhibited less pronounced changes in their chemical signature. Their volatile fingerprint is characterized by upregulated production of esters and 2-ethylhexanol and downregulated production of other alcohols. The results obtained within this study provide evidence that gastric cancer alters the volatile organic compounds profiles of cell lines under study. The components of this fingerprint secreted from human organism via breath could assist in the non-invasive diagnosis of gastric cancer, employing sensitive mobile platforms. Whether the markers found within this study might have a value as biomarkers for gastric cancer diagnosis remains to be clarified in future studies. This work was supported by Fundamental and Applied Research Project Program in Latvia, Volatile Organic Compound for Potential Application in Gastric Cancer Screening, and by European Union's Horizon 2020 Research and Innovation Program. The authors do not declare any conflict of interest, and thank you very much for your attention.
My name is Donato Altomare. I'm full professor of surgery at the University of Bari, Italy. I'm presenting a poster entitled Chemical Signature of Colorectal Cancer Case Control Study for Profiling the Breath Print. Colorectal cancer is among the 4 biggest cancer killers in Europe countries, and the availability of an effective screening test can improve early diagnosis and survival. Recently, breath testing has been pursued as an option for colorectal cancer detection, and a range of excellent volatile organic compounds with discriminatory ability have been identified. The aim of this case control study was to evaluate whether the use of a new advanced breath sampler, receiver breath sample by Holstone, able to capture only the alveolar fraction, excluding the environmental contaminant, can improve the reliability of breath testing for colorectal cancer screening and early diagnosis. This study recruited patients of any sex or age undergoing curative surgery for histologically proven adenocarcinoma of the colon or rectum of any clinical stage, and a comparative group of non-cancer control who had a negative colonoscopy. Once collected in the receiver sorbent tubes, the VOCs were processed by a thermodissolver. Products were connected directly to an heater transfer line and then to the gas chromatograph mass spectrometry equipment. To assess the accuracy of the discriminatory ability of VOCs for the detection of a patient with colorectal cancer, the analysis was conducted in 2 stages. The first stage is constructed a receiver operating characteristic curve for each VOC, and with the establishment of an individual cutoff value for discrimination of colorectal cancer. The second stage involved cross-validation using the leave one out method. As you can see in the table, 89 colorectal cancer patients and 90 non-cancer controls entered the study. 38 patients were in clinical cancer stage 1 and 2, and 42 in stage 3 and 4. The groups were matched for sex, but the mean age of patients with colorectal cancer was significantly higher compared with the controls. Hypertension was the only comorbidity which was more common in patients than in controls. Using the relevant ROC curve from sensitivity, specificity, and true predictive value with a cross-validation univariate analysis, the VOCs were ordered from the most to the least accurate. 15 VOCs were found to be statistically significant in discriminating colorectal cancer patients from controls in univariate analysis as shown in Table 2. The highest area under the curve values were noted for tetradecane, ethylbenzene, methylbenzene, octanol, nonanol, and decanol. We also obtained a multivariate models. In this figure, you can see 3 models comparing cancer and non-cancer patients. Model A was set up with adjustment and no adjustment of age, as you can see in figure A and B. The model showed a significant likelihood of association, confirming a 90% rate of true positive and 93% of true negatives, and a predictive value of 93%. In the model B, there was a significant likelihood of discrimination for early stage colorectal cancer compared with controls, and the model C compared advanced colorectal cancer with no cancers and showed a significant likelihood of discrimination, as you can see in figure D. In conclusion, the pattern of VOCs identified showed a very good reliability in discriminating cancer patients from healthy controls, irrespectively of the cancer stage. Confirming this pattern of VOCs in a multi-center international study might support the next step in clinical application of breath sampling in colorectal cancer, which is to create a smart, inexpensive, online sensory device to be used for mass screening of colorectal cancer, driving patients to further investigations such as a colonoscopy. Thank you for your attention.
Hi everyone, my name is Robyn Larracy and I'm a master's student in biomedical engineering at the University of New Brunswick in Canada. We've been collaborating with the Horizon Health Network and Picomol, a medical technology company specializing in breath-based health solutions, to develop a lung cancer screening system using infrared cavity ring-down spectroscopy. Lung cancer remains the most prevalent and most deadly form of cancer worldwide. Early detection is critical to catch the disease in its early treatable stages, but nearly 80% of cases are discovered when the cancer has already begun to spread. So in this study, we've considered a novel screening system based on the analysis of exhaled breath samples using cavity ring-down spectroscopy. We've used machine learning to identify potential biomarkers and to develop a classification system for differentiating lung cancer and non-lung cancer individuals. 10-liter alveolar breath samples were collected onto 10x TA sorbent tubes for 96 control subjects and 62 pre-treatment non-small cell lung cancer patients. Collection was done using Picomol's proprietary breath sampler, which is pictured here on the slide. Participants with a variety of other lung conditions, so including COPD, asthma, pneumonia, and bronchitis, were included in both groups, 36 control subjects and 44 lung cancer subjects. This table of clinical factors and demographics shown here is also on the poster if anyone is interested in taking a closer look at that. Each of the collected samples was analyzed with CRDS at 4 different desorption temperatures, 75, 150, 225, and 300 degrees Celsius, yielding 4 spectra for each subject. Pictured is an example of a 75-degree spectrum, which you can see consists of 73 CO2 wavelengths, which are in the mid-infrared region. We then applied a stepwise spectral fitting algorithm to identify the VOCs presented in each of the spectra. The fitting program finds the linear combination of compounds that provides the best fit from a reference library of 152 VOC spectra. For the example spectrum shown, the model identified and quantified 15 compounds, shown here to the right of the spectrum. These VOC concentrations were used to create the classification system. First, the concentrations were transformed. So we considered various logarithmic transformations as well as a square root transformation and a cube root transformation. Next, the top VOCs were selected using minimum redundancy, maximum relevance feature selection, which ranks features sequentially based on a difference of Pearson correlations. So that's the feature's correlation with the group membership labels minus its correlation with other features. Linear support vector machines were then trained and validated using leave one subject out cross-validation to evaluate these top sets of MR-MR ranked features. Of the 6 different transformations we considered, the best cross-validation performance was achieved using the natural log transformation with 30 features. This model had an accuracy of 85.44%, sensitivity 77.42%, specificity 90.63%, and an area under the curve AUC of 0.858. 4 VOCs appeared twice in the feature set from the different desorption temperature spectra: isopropanol, trifluoromethane, tert butyl alcohol, and ethyl tert butyl ether. Additionally, 8 of these top selected VOCs have been found to differ between lung cancer and control subjects in other works, including dimethyl sulfide, isopropanol, and undecane, though many of these studies include small cell lung cancer subjects, which we've excluded from our study. These preliminary results show promise for a novel exhaled breath-based screening system using infrared CRDS technology and spectral VOC fitting. We have previously built a model for this data using spectral patterns as classification features rather than VOC concentrations, which achieved similar accuracy. Future work will compare these 2 approaches. I would like to acknowledge our funding partners, the New Brunswick Health Research Foundation and MyTax, as well as all of the clinicians, research coordinators, and nurses that have contributed to sample collection at the St. John Hospital, Dr. Everett Chalmers Hospital, and the Moncton Hospital. Thanks, everyone.
My name is Bruce Johnson, and I'm a physiologist at Mayo Clinic. And the title of our project is Breath Biopsy of Elite Runners Engaged in Exhaustive Exercise. And this was a collaborative project with our colleagues at Owlstone, as well as colleagues at the Ultra Sport Science Foundation in France and the National School of Sport and Alpine Skiing in Chamonix, France. As a bit of background, our laboratory has had a long history of studying the limits of human performance and adaptation in extreme environments. And in particular, we've been interested in the respiratory system and how this is involved in adaptation or maladaptation in these extreme conditions. This particular project was focused on examining the array of volatile organic compounds that occur in the exhaled breath before and after an exhaustive ultramarathon. So if you look at figure 1, this just shows the Ultra Trail Mont Blanc. So people have to compete to get in this, and so they are considered some of the best ultra trail runners. We were interested in Chamonix and the Alps because of the altitude changes and how that might affect some of our results. And again, we studied individuals before, immediately after, and in some people during the recovery phase. We used the Owlstone platform for capturing VOCs. And we did more or less a pilot study in 2018. During that study, we had saturation of one VOC particular that limited our ability for further analysis. And so in 2019, we went back planning in a different strategy for analysis. We were able to collect data on 29 volunteers pre-race, 24 post-race, and so we had paired samples in 24 subjects. Table 1 just shows some simple demographics, 3 women and 21 men, average age late 30s. Going down to our results, I'll read some of this from the poster. But basically, across all samples, a total of 811 different VOC signals were identified, which was a significant improvement over 203 identified in 2018. Of these identified, 793 were detected with suitable frequency to enable their use for further analysis. We used the Breath Biopsy HRAM library of verified VOCs, which allowed 74 of the 811 features to be assigned and verified for identity. I'll go over figures 3 and 4 quickly. These are principal component analysis over the entire data set to identify sources of variation. They are color-coded, and also 4 represent pre- and post-race data. But basically, the plot of PC1 and PC2 demonstrates a trend in post-race sample composition, and PC3 and PC4, separate pre- and post-race samples, a similar somewhat trend. This essentially indicates effects related to the race are not the most important or significant source of variation across all samples, but they are a key factor. I think figure 5 is the most interesting plot. This just shows fold changes with 63 features that showed the most significant differences between pre- and post-race, 51 increased, 12 decreased. And of particular interest was one signal that had a 59-fold increase post-race, that's figure 6. And interestingly, this was the same peak that caused saturation problems in the initial study. Figure 7 just shows a couple additional molecular features and their abundance pre versus post race. To conclude, collected matched pairs of pre and post race samples from 24 athletes during the 2019 Ultra Trail Mount Blanc race were assessed. Enhanced study design analysis method provided better sensitivity and dynamic range, resulting in extraction of 811 features, making it possible to measure a superabundant peak that previously confounded the analysis, but this time was actually identified as acetic acid. 161 features showed a significant difference between pre and post race, with 63 retaining their correction from multiple testing. And of these, majority, 51, again, increased post race. We are exploring further what the superabundant peak, the source of the acetic acid might be, and examining the other VOCs to determine how they relate to our measures of physiologic stress and inflammation, and more specific measures of the respiratory system. So we look forward to presenting additional data in future symposiums and opportunities for presentation. Thank you.